get started, folks. Skeletal muscles are innervated by nerves of the somatic nervous system. The axons which provide innervation to the muscle are carried by a nerve. So this nerve is going to have multiple axons. And as these axons approach the muscle, they begin to branch. So you see that this axon then branches perhaps into 3, 4, sometimes some axons branch into hundreds of branches. Okay? And at the end of each axon branch, okay, it forms a neuromuscular junction. Here we see a neuromuscular junction, the junction between the neuron and the muscle cell. Okay, notice that the end of this axon branch, we can call it an axon terminal. Notice that it doesn't actually touch the postsynaptic membrane of the muscle fiber or cell. That there's a space between the two. Okay? That's called a synaptic cleft. So there's a space, a synaptic cleft, between the presynaptic membrane from the axon terminal and the postsynaptic membrane of the skeletal muscle cell. Now notice also that at the site of the neuromuscular junction, the postsynaptic membrane of the muscle cell is folded. This can be called a motor end plate. So the neuromuscular junction is going to consist of an axon terminal, a synaptic cleft, and a motor end plate along the postsynaptic membrane of the muscle cell. Okay. Now, make this bigger. So again, this slide is just a reiteration of what we said here. It says the neuromuscular junction is formed by axonal endings, or I can call this an axon terminal, okay, which have small membranous sacs inside. We've talked about these before. We call these synaptic vesicles. These little sacs house the neurotransmitter that's going to be released from that presynaptic membrane. In this case, for somatic motor neurons, that neurotransmitter is acetylcholine. It goes on to say that the motor end plate of a muscle is the specific part of the sarcolemma that contains acetylcholine receptors. So this little motor end plate here is going to have receptors for the acetylcholine. So that when acetylcholine is released, it's going to diffuse across the synaptic cleft and bind to those receptors. Okay? And it goes on to say that although exceedingly close, the axon terminal and the motor end plate don't actually touch. They're separated by this space called the synaptic cleft. And this kind of demonstrates a space-age view of the neuromuscular junction. Here you have the axon terminal here. You can see here the synaptic cleft, and the folded part of the sarcolemma is called the motor end plate. In this magnified view here, okay, of course, this is the presynaptic membrane. This would be the postsynaptic membrane. Okay. You can see that these synaptic vesicles are going to fuse with the presynaptic membrane, and in doing so, they're going to release acetylcholine into the cleft. Acetylcholine can then bind to acetylcholine receptors. These would be nicotinic M receptors.
Is it folded just for more surface area?
Yeah, to increase surface area. Now, notice that these acetylcholine receptors, these nicotinic receptors, are linked to voltage-gated channels. I'm sorry, chemically gated channels. Such that when the chemical, the neurotransmitter, binds the receptor, it opens up these chemically gated channels to allow sodium to come into the cell. That's what's been shown here. At the same time, it allows the egress of potassium. Now, it turns out that more sodium enters than potassium leaves. So as sodium enters into the cell, it's going to result in a change in membrane potential. It's going to make the interior of the cell more positively charged. It's going to bring about depolarization. And if there's strong enough depolarization, then that wave of depolarization can spread down the sarcolemma. As we pointed out before, there's an enzyme located within this neuromuscular junction called acetylcholinesterase. Its purpose is to break down acetylcholine so that it can continue to stimulate the receptors on the sarcolemma, on the motor end plate. Okay. Everybody with me? This is another kind of space-age view of the muscle. Let's see if y'all can figure out what's going on here. This, of course, is an axon terminal. This is the motor end plate. What are these structures called here?
The synaptic vesicles.
Synaptic vesicles, that's correct. Now, what do you think this is here within the mitochondria? What is this white membranous structure?
I know down the middle is the T2.
Well, right. This entire thing is the sarcoplasmic reticulum, and these cross channels on either side of the T-tubule, these are terminal cisternae. The pair of terminal cisternae with the T-tubule make up one of those triads. Remember that the T-tubule is simply an inward extension of the sarcolemma, so that as a wave of depolarization passes along the sarcolemma, it can pass down into the cell through the T-tubule. And this slide goes through the process of muscle stimulation. It says when a nerve impulse, another word for that is an action potential, when it reaches the end of an axon at the neuromuscular junction, voltage-gated calcium channels open. Okay? These are voltage-gated calcium channels along the axon terminal here. All right, so when an action potential reaches the end of the axon and it opens up these channels and allows calcium to flood into the cell, it's the ingress of calcium that causes the synaptic vesicle to fuse with the presynaptic membrane. Okay? This calcium inside the axon causes axonal vesicles, synaptic vesicles, to fuse with the axonal membrane. This fusion releases acetylcholine via exocytosis. Acetylcholine is going to then diffuse across the synaptic cleft, and it's going to bind to receptors, nicotinic M. Okay? That triggers opening of chemically gated channels that allow sodium to come into the cell, and it's going to trigger an action potential. Okay? Acetylcholine is quickly destroyed by the enzyme acetylcholinesterase. This prevents further stimulation of the muscle. Okay. Again, a reiteration of that which we've been talking about. Acetylcholine binds the nicotinic M receptors. It opens chemically gated channels, sodium comes into the cell, and the sarcolemma becomes less negative. This is called depolarization. When it spreads like a wave, we call that an action potential. So it says an action potential is a transient depolarization event that includes polarity reversal of the sarcolemma. It means that the inside goes from being negatively charged to positively charged, and then it propagates along the sarcolemma. Remember, this is the situation at rest, where sodium is concentrated mostly on the outside, which has a positive charge. The inside of the cell has a negative charge. Okay? The cell interior, its major electrolyte is potassium instead of sodium. But as a result of the differential in ionic composition, the outside of the cell has a positive charge with respect to the inside. This is called the resting membrane potential. Now, we said with the opening of chemically gated channels, sodium comes into the cell. That means at the site where sodium enters, we have a change in membrane potential, such that the inside now becomes positive. Okay? The resting membrane potential is decreased. Depolarization occurs. If there's enough sodium that moves into the cell, then this depolarization event is self-propagating. It's going to start to move across the entire sarcolemma. Remember, what happens is when sodium comes in, it's attracted to the negative charges along the adjacent patch of membrane. So as sodium moves, if enough of it moves to the adjacent segment, it's going to open voltage-gated sodium channels that allows more sodium to come in, and it propagates this wave. Now, when sodium comes in, the membrane depolarizes. That opening of voltage-gated sodium channels is going to be followed by the opening of voltage-gated potassium channels that allows potassium to leave and repolarization to occur. So on the heels of the opening of the voltage-gated sodium channels, you have the opening of voltage-gated potassium channels, which bring the membrane back to the original resting membrane potential. We studied this when we first started talking about the nervous system. It's the same sequence of events. Now, once that action potential is generated, it not only spreads down the sarcolemma, but it travels down those T-tubules. When it reaches the triad, in other words, the region where the T-tubule is surrounded by the terminal cisternae, it's going to trigger release of calcium. You know what happens when calcium is released. JJ played the role of calcium. Calcium attached itself to troponin. Who played troponin? Alex, you were the mother. And once calcium binds to troponin, troponin changed its shape, pulling away tropomyosin from the binding sites on actin. So the actin binding sites became exposed. Okay. At that point, myosin all of a sudden formed the cross bridge with actin. Why? Because there was an affinity and the binding site was exposed. Okay? And remember what happens during muscle contraction. There's a series of ratcheting type events where there's attachment, power stroke, release, cocking, attachment, power stroke, release. Okay? And as a result, thin filaments move toward the center of the thick filaments. Okay? Now, what powers this is ATP. Every time ATP attaches to the myosin head, it triggers its release and cocking. Okay? It stores energy in the head so that when it again attaches, it performs the power stroke. Okay? Now, it actually pumps along the membrane of the sarcoplasmic reticulum, start to pick up that calcium and move it back into the sarcoplasmic reticulum. Those pumps are active transport pumps. They're fueled by ATP. Once calcium undergoes reuptake back into the sarcoplasmic reticulum and is no longer binding to troponin, then the tropomyosin then recovers the active binding sites. The muscle relaxes. And this slide kind of shows everything in one slide. It shows how the binding of the acetylcholine to the receptors triggers an action potential that spreads down the T-tubule. It brings about release of calcium. Within the sarcoplasm, calcium is going to bind to troponin. In doing so, it's going to displace tropomyosin that allows the myosin head to bind to the actin. Okay? So this is going to initiate the sliding of the filaments. Now, in the meantime, calcium undergoes reuptake back into the sarcoplasmic reticulum. When that occurs, then tropomyosin again recovers the binding site and muscle contraction ends. The muscle relaxes. All right, so this is the sequence of events. Cross-bridge formation is simply when the myosin head becomes bound to the binding site on the actin. Okay? Remember that when the myosin head attaches, it's already cocked. Once attachment or cross-bridge formation occurs, then we initiate the power stroke, where the myosin head pivots, and in doing so brings the thin filament in closer to the center of the thick filament. Now, if ATP is present, it attaches to the myosin head so that it can then detach, and the cycle begins again. Once it detaches, it cocks, it attaches again, forms a power stroke, and we get the whole series of events recurring. cocking of myosin head is a result of hydrolysis of ATP. In other words, the energy derived from cleaving one of those high energy bonds is used to cock the head of the myosin. Okay? We increase its potential energy. With the power stroke, that kinetic energy is transferred into, that potential energy is transferred into kinetic energy. All right, just some terms. When we use the term contraction, we're referring to the activation of those cross bridges. Okay? Shortening occurs when tension is generated. The tension generated by the cross bridges exceeds the forces opposing shortening. Contraction ends when the cross bridges become inactive. Okay? And the myosin heads detach. All right, let's go back to a basic concept. Notice that a nerve, this could be, if this is the biceps muscle, the nerve that innervates the biceps is the musculocutaneous nerve. It may be carrying 100 different axons that are providing innervation to the individual muscle cells within the biceps. Okay. Now, a motor unit is a motor neuron and all the muscle fibers it supplies. So for example here's an axon of a single neuron, but because it branches 3 times, it's providing innervation to 3 different muscle fibers. Okay? So that motor unit here is providing innervation to 3 fibers. Okay? Now, typically, the number of muscle fibers per motor unit varies from about 4 to several hundred. Okay. Muscles that control fine movements, like the muscles of the hand or the muscles of facial expression, have very small motor units. In other words, each neuron or axon is only providing innervation to maybe 3 or 4 or 5 different muscle fibers. On the other hand, large weight-bearing muscles like the gluteus max, okay, or the hamstrings, or the quadriceps muscle, they have large motor units where a single axon or neuron may provide innervation, may branch numerous times, innervating, let's say 100 or 200 muscle fibers. Okay? So these large weight-bearing muscles provide bulk movement. Smaller muscles, which are capable of precision control, they have much smaller motor units. Much smaller motor units. Now, the muscle fibers from an individual motor unit, let's say with this axon here, these axons are not going to provide innervation to 3 fibers, let's say, on the surface. The fibers that are innervated by a motor unit are spread throughout the muscle. Okay. Contraction of a single motor unit is going to cause weak contraction of the entire muscle. And here's some examples here. We showed 2 different motor units. Again, here's a nerve that's carrying 2 axons. This axon is branching and providing innervation to 3 fibers. Here's another axon that's branching, in this case is innervating 2 fibers. Now, let's talk a little bit about muscle physiology. A muscle twitch is a response of a muscle to a single brief threshold stimulus. When I was in college, I was in a physiology lab where we took tiny muscles from a frog. Okay. We stretch a strip of muscle across this device here. I hold it in place. And we could apply little jolts of electricity to the muscle, a little electrode, that caused it to twitch, to contract briefly. Okay? And that twitch consists of 3 different phases. There's a latent phase, and that's the interval between stimulation with an electrical impulse and the beginning of muscle shortening. Okay? Then there's the period of contraction. That's when the muscle actively begins to contract and shorten. And then it's followed by a period of relaxation. This is the period in which calcium is moving back into the sarcoplasmic reticulum, and the muscle tension falls back to baseline. And in fact, you can record the shortening. These little devices here that are holding the muscle in place can lead to recording on a graph, like we demonstrate here, where this interval between the onset of the stimulus at the beginning, let's say time 0, and the beginning of muscle contraction, this is called the latent phase. Okay? Then you have the period of contraction here, where the tension builds up within the muscle, and it's followed by a period of relaxation. This is called a myogram. Okay? This latent phase is simply that period of excitation, contraction, coupling. That latent phase is that interval where an action potential is traveling along the sarcolemma, then down the T-tubule, calcium's being released. Calcium then is binding to troponin, okay? And then finally, when tropomyosin is displaced, the myosin head then forms a cross bridge. All of that activity is occurring during the latent phase. It's only then when the cross bridge forms and begins to bring about the power strokes that we see shortening. Now, muscle responses are graded. In other words, if I want to pick up this marker and put it in my pocket, okay, I don't do this and drop it, okay? In other words, I don't apply all or none. You know, all force, maximal force, and then none. No, responses are graded. I apply just enough strength, tension, to pick up the pen, to move it at a speed appropriate to reaching my pocket in a coordinated fashion, and then just kind of slowly let it drop into the pocket. So, muscle contraction is modulated. Okay? Graded responses provide variation in the degree of contraction, and they're required for proper control of skeletal movement. Now, how do we grade responses? Well, grading is determined by 2 things. Number 1, the strength of the stimulus. The strength of the stimulus determines how many motor units are going to be involved in that muscle contraction. Stronger stimuli involve more motor units. When you have more motor units involved, then more muscle fibers contract. You also can change the strength of the stimulus by increasing the frequency of action potentials traveling down an axon. So before, I showed you a nerve. I said that nerve is carrying multiple axons, each of which is branching and providing innervation to individual fibers within the muscle. Okay? I can change the strength of a stimulus by involving more motor units, or with respect to an individual muscle motor unit, sending down impulses more frequently, increasing the frequency of stimulation. Okay? As more impulses reach the axon terminal, more impulses per second, more acetylcholine is released. As more acetylcholine is released, more chemically gated sodium channels open, more sodium comes into the cell, okay, and it ensures generation of more action potentials.
Is that something we like do ourselves, think about it, or?
Well, in most cases you don't think about it. In most cases it's subconscious.
Yeah, okay, that's what I was.
It's a learned behavior. In other words, you know that if you want to pick up this book, you're going to apply a little bit more than this.
So do you have like a disease or a disorder that messes with your graded muscle responses?
Oh, you certainly can. For instance, myasthenia gravis is a condition where you form antibodies against those acetylcholine receptors. so that even though you want to generate a certain response, a certain degree of contraction, you can't. You have muscle weakness. Now, how does increasing the frequency of stimuli augment the motor response? Okay? This shows a single twitch, the response you would get from a muscle or muscle fiber. Okay? If you began to deliver stimuli more frequently down that same axon, then the muscle wouldn't have a chance to completely relax before the next stimulus comes in, or then before the next stimulus comes in. And so with each new stimulus, more calcium is going to be released before the previously released calcium gets reuptaken, if that makes sense. So more calcium is going to be left within the sarcolemma to generate a stronger degree of muscle shortening. So it says frequently delivered stimuli, in which the muscle doesn't have a chance to completely relax, increases contractile force. This is called wave summation. Okay? Again, it's part of the reason is that with this contraction, if it were to completely finish like this, the reason that this is relaxing fully is because the calcium undergoes reuptake back into the sarcoplasmic reticulum. In this case, before all the calcium undergoes reuptake, more calcium is released. So that it's beginning to build up within the sarcoplasm. Okay? As more calcium is released, there's more calcium available to bind to troponin, to expose binding sites. Okay, which can result in a stronger degree of contraction. If we release stimuli even more frequently, if we deliver stimuli even more frequently, then we're going to get incomplete tetanus, where with each new stimulus, the degree of contraction becomes progressively stronger until we reach a point which is called tetanus, sustained muscle contraction.
So could you give me an example where the muscle does not have time to completely relax, like an exercise, basically?
All right. If I'm doing this and I'm applying maximal pressure, okay, this is in effect tetanus. This is sustained muscle contraction, okay, where I'm delivering stimuli at a frequent enough interval that the muscle doesn't have a chance to relax, okay? And what I'm saying is when you go from 0 to 100, you're delivering stimuli at progressively greater frequencies to generate stronger and stronger degree of contractions. Now, let's talk about strength. We talked about frequency, that by delivering stimuli at greater frequencies, we can result in an increased strength and contraction. But contraction is also related to stimulation strength, the number of motor units involved. The threshold stimulus is a stimulus strength at which the muscle first begins to contract, in which we have an observable muscle contraction. Beyond threshold, muscle contracts more vigorously as stimulus strength is increased. Or another way of saying that is as more motor units are recruited. Okay? You're bringing more muscle fibers into play. So that if you look at this bottom diagram, if only one motor unit is involved, you're going to get a certain degree of tension forming. If a second motor unit is involved, more tension. A third, even more, until you get all the motor units recruited. Okay. Again, we talked about tetanus. That's a sustained muscle contraction. Now, when you're sitting down at your desk at rest and maintaining a certain posture, okay, that partial contraction of the muscle that's occurring right now on an ongoing basis, this is called tone. In other words, when you're sitting, you're not contracting those paraspinous muscles maximally, but partially, okay? And this partial contraction, this tone, is generated by spinal reflexes, okay, that activate first one group of motor units, okay? So these motor units then cause contraction of some of the muscle fibers, and as they contract, then it triggers an impulse that then lets them relax at the same time that another motor unit is being recruited. Okay, that's why you don't fatigue. Is that there's a continuous relay of sensory and motor impulses back and forth from the spinal cord that's first triggering contraction of some of those muscle fibers, by some motor units. And then as sensory reflexes pass back to the spinal cord, it then lets those relax as it's at the same time recruiting other motor units. So at no one time are all the motor units firing, but some are firing at all times.
Right.
So that's the ones that are firing right now, then the next second they're relaxing.
So each of them like take breaks?
They take turns and breaks, so that way you don't fatigue. Now, if someone has, let's say the innervation to a muscle is completely cut, okay, so that there's no transfer of impulses back and forth to the spinal cord, then that person's going to lose all muscle tone in that muscle. Okay? It would be equivalent to having a lower motor neuron injury. You're going to lose all muscle tone. And it's pretty easy to determine if all muscle tone has been lost. If you have weakness in this arm, okay, and let's say you have a great deal of weakness, but you haven't lost all muscle tone, I can put this hand up here, and if I let go, she may let it fall, but it's not going to hit her head. She's got enough tone in there that as it falls, it'll kind of avoid her head. On the other hand, if she has no muscle tone and I let go, what's it going to do? It's going to hit her in the head. Okay? Now, when we do exercise, what do we like to do? If you ever ran track or field, what do you do before you start?
Stretch.
Stretch. You warm up. So before you do a sprint, before you do your 200, you may do a few strides up and down the football field, right? What you're trying to do is elicit the trepe effect. It's also called the staircase effect. This is increased contraction in response to multiple stimuli of the same strength. I'm going to come back to this slide. In other words, when you keep sending impulses down to a muscle, even though the impulses are of the same strength, over time we see a warming up effect, where the contraction response to that same single stimulus begins to increase. It's a warming up effect. Okay? And the reason we see this warm-up effect, or this staircase effect, is because there's more calcium available in sarcoplasm, and the enzyme systems, like the enzymes that break down ATP in the myosin head, okay, they warm up. They become more efficient. So that in response to the same stimulus, you get a greater effect. This is a warm-up effect. Okay, we talked about tone. Tone is the constant, slightly contracted state in all muscles. It doesn't produce active movement, but it keeps the muscle firm. Okay, it keeps it healthy and ready to respond. Spinal reflexes are responsible for this tone by first activating one motor unit, then another. They respond to activation of stretch receptors by relaxing that muscle unit and then activating another. Okay. Now, we differentiate between isotonic contractions and isometric. Isotonic contractions are ones that result in a change in muscle length and moving a load. For instance, if I'm lifting a weight like this, and in lifting the weight, I'm moving it, okay? And I'm causing the angle of this joint to decrease. That's an isotonic contraction. The muscle changes in length. This biceps muscle shortens as I contract the muscle. Now, I showed earlier an example of isometric contractions, where I'm contracting muscles, but they're not shortening. Nothing's moving. Okay? That would be referred to as an isometric contraction. The muscle neither shortens nor lengthens. This occurs if the load is greater than the tension the muscle is able to generate. Okay. Now, finally, let's talk about development of energy for muscle contraction. You know that muscle requires ATP. We need ATP. Why? ATP is going to detach the myosin head. It's going to be responsible for cocking of the head. In other words, and that's generation of potential energy. And then when that head attaches to the actin, it converts that potential energy to kinetic energy with the power stroke. So that movement, that ratcheting, is all in effect, is all the result of generation of energy from ATP, the hydrolysis of ATP. ATP is also needed to pick up the calcium and put it back in the sarcoplasmic reticulum. Remember, that's an active transport pump. Let me ask you a question. Why is it that when we die, we develop rigor mortis? In other words, the muscles become contracted and stiff.
Because there's no ATP for calcium for like to be reuptaked or whatever.
Right, for calcium reuptake.
For calcium reuptake.
I mean, we're
So it
dead.
stays
So
stiff.
the muscle doesn't relax.
Yeah.
And what's another reason? Well, you need ATP to cause detachment of the myosin from actin, which begins relaxation. And if you don't have ATP to attach to the myosin head, then the actin and the myosin won't detach and relax, bring about relaxation. So when you die, you can't generate new ATP, and so the muscle can't relax. It remains stiffened and contracted until the muscle begins to break down. That makes sense, everybody? Okay. Now, the muscle stores some energy. The muscle will store enough ATP for about 4 to 6 seconds of activity. Once that ATP is used, it's got to be regenerated. Okay? It's regenerated by 3 processes. One is the interaction of ADP with creatine phosphate. Creatine is a muscle protein that can create a high energy bond with phosphate. In other words, creatine can form, and when I use this little wavy line, it means a high energy bond. It's a bond that's easily broken, and when broken, releases energy. Okay? We said that muscles can store up to 4 to 6 seconds worth of ATP. ATP stands for adenosine triphosphate. Adenosine triphosphate, with these last 2 bonds being high energy bonds. And really, when we say high energy, we really mean that they're easily broken, and when broken, release energy. Now, we store about 4 to 6 seconds worth of this ATP. Okay? When we use the energy stored in the ATP, we're really breaking this bond here and converting ATP to ADP and phosphate. Does that make sense? In other words, we're losing one of these phosphate groups.
Because ADP only has 2 things.
ADP only has 2.
Right.
All right, so how do we reform or regenerate ATP? One way is by taking this ADP and reacting it with creatine phosphate. We can transfer this high energy bond to ADP, and in doing so regenerate ATP. Yeah, and then creatine. Okay? So one way to regenerate ATP is through interaction with creatine phosphate. This is called, by the way, there's a name for this. It's called substrate level phosphorylation. In other words, we're simply transferring this phosphate group from one molecule to another. We're regenerating ATP that can be used then by the muscle to generate contraction. Now, the other way we generate new ATP is through anaerobic glycolysis. We've talked about this already. Anaerobic glycolysis is a process where we take glucose and we break it down in the sarcoplasm without the use of oxygen. We take glucose, and through a series of about 10 steps, we convert it to 2 molecules of pyruvate, each of which has 3 carbons. This glucose has 6, so we conserve the carbons. but this metabolic pathway generates 2 molecules of ATP per glucose. Okay? It does not use oxygen. Now, if oxygen is available, then we can carry out aerobic respiration, where we take the pyruvate, transport into the mitochondria, okay, and carry out the Krebs cycle. In this case, the pyruvate inside the mitochondria is going to be converted to acetyl coenzyme A, and that then is going to participate, that's going to be able to then enter into this Krebs cycle, a series of reactions that generates about 36 molecules of ATP. This Krebs cycle, this aerobic respiration, requires oxygen. That's why it's called aerobic. Glycolysis is anaerobic. It doesn't require. So these are different mechanisms by which muscles can generate ATP to continue to contract. In this diagram, these different mechanisms are represented by these 3 different columns. This is substrate level phosphorylation, where we take ADP and it picks up phosphate from creatine phosphate, generates ATP. Here, glycolysis, where we take glucose, break it down into pyruvic acid. Now, if oxygen is not available, that pyruvate gets converted to lactic acid. It brings about muscle fatigue.
Is that where your muscles start to cramp?
Yeah, that's part of the buildup of lactic acid. Okay. Now, if oxygen's available, though, pyruvate then enters into the mitochondria, and we can generate a whole lot more ATP. Okay. Waste products, though, include CO2 and water. This is aerobic metabolism. Okay. Now, with substrate level phosphorylation, it's not very efficient. We only generate 1 ATP per creatine phosphate. But we have enough creatine phosphate that by this mechanism alone, we can provide energy for muscle contraction for up to 15 seconds. But then we're going to run out of creatine phosphate. Okay? What about anaerobic metabolism? It doesn't use any oxygen, but it can provide energy for up to 30 to 60 seconds, up to a minute. So if you're running the 400 meter, that might be enough. But at the end of that period, lactic acid is going to begin to build up, and your muscles are going to fatigue. So you'll notice at the end of that 400 meter race, you're starting to tire out. Your muscles are beginning to fatigue. You're slowing down. On the other hand, through aerobic mechanisms, Krebs cycle, okay, you can supply a lot more energy, a lot more ATP that can power the muscles for hours. Okay, and again, it's with the supposition that the amount of exercise you're doing is not exceeding the ability to provide oxygen to the muscle. In other words, the demand doesn't exceed the supply.
So wait, like these 3 systems, does your body choose, or it just?
No, if you have plenty of oxygen available to the muscle, all 3 are occurring.
Okay.
Okay.
So it just depends on what.
But let's suppose you're doing really maximal strength exercise,
okay,
like a sprint.
Okay.
Well, a sprint's the muscles are gonna demand more oxygen than can be supplied. In which case, then you're going to be limited to anaerobic mechanisms,
right?
Because the muscle demand is going to outstrip the supply, and you won't be able to carry out aerobic. On the other hand, if you're running a marathon, where you have continuous muscle activity, but it's not maximal muscle activity, then you're going to continue to be able to supply those muscles with oxygen,
right? Because
the demand is not as great. It's not maximal muscle activity.
So it just depends on the environment that you're in.
Right, so that's why in a marathon you can continue to run for hours, because you're capable of generating ATP through aerobic mechanisms. Okay? So, when muscle contractile activity reaches 70% of maximum or more, okay, then aerobic mechanisms like the Krebs cycle can no longer sustain muscle contraction. One thing, You notice when you're exercising that the muscles are beginning to bulge, right? Have y'all seen that? You have a look in the mirror when you're at the gym? People like to see their muscles are beginning to get bigger.
Yeah.
Okay? As they do so, they compress blood vessels that deliver oxygen. Okay? So that impairs delivery of oxygen. Okay? So with 70% or more of muscle activity, you begin to limit the supply of oxygen. And when you limit the supply of oxygen, that pyruvic acid that's generated during anaerobic metabolism begins to build up. It has no place to go. It can't enter into the Krebs cycle because you need oxygen for that. Instead, it's converted to lactic acid. And as that lactic acid builds up, your muscles begin to fatigue. Now, the lactic acid is going to leave the muscle cells and enter into the bloodstream. Okay? It's going to be picked up and can be used as fuel by different organs. In the liver, it can be converted back to pyruvate. Muscle fatigue, that's the feeling at the end of the 400 race. Okay? The muscle's in a state of physiological inability to contract. Okay? In other words, the spirit is willing, but the body is weak. You know, you're bench pressing 200 pounds, and once you get to 10, all of a sudden, you're starting to slow down. You can't quite make it, that number 11 or number 12. Your muscles are fatiguing. That's because ATP production is starting to lag behind. It can't keep up with use. You develop a deficiency of ATP. Okay? Along with that, okay, lactic acid is beginning to build up. As it builds up, that muscle is going to fatigue. You also develop imbalances of electrolytes that contribute to the muscle fatigue. Now, you notice that if you stop, whatever it is you're doing, it takes a while for the muscles to recharge. Okay? That vigorous exercise, whatever you did that led to fatigue, causes dramatic changes in muscle chemistry. So that to recharge the muscle so that you can start all over again, start afresh, the muscle has to return to its resting state. The oxygen reserves have to be replenished. Okay? Lactic acid, okay, must be converted back to pyruvic acid. You have to get rid of all that lactic acid. Glycogen stores have to be replaced. ATP, creatine phosphate reserves have to be restored. And so what happens is the muscle develops an oxygen debt. That's the amount of oxygen that's required to restore oxygen, to restore pyruvate, to restore ATP and creatine phosphate. It takes time. Now, turns out that only 40% of the energy released in muscle activity is used for work. The other 60%, okay, is released as heat. So when you're actively contracting muscle, you're generating heat. And that heat has to be dissipated, usually as sweat. Okay? So when you see all these people that are running in the Crescent City Classic, they're sweating, they're drinking water to replenish the water that's lost in sweat. They're also pouring the water on them to help dissipate that heat. Now, I'm not going to go through the different muscle fiber types, but starting next class, today we're going to look at microscopes, but starting next class, we're going to start reviewing the individual muscles. Now, next class, we're going to review the muscles of the axial skeleton. That includes the muscles of the head and neck, the face. It'll include some muscles of the trunk. And then in the classes that follow, we'll talk about the muscles of the upper and lower extremities. I would really encourage you, if possible, to read ahead, because the names are going to sound very foreign. A lot of them are Latin-based. And if you've read the section already, then it tends to be easier to pick up the material. It's not tough. It's that all the words sound foreign. Okay? When we talk about the muscles, what you'll be expected to learn is the origin and insertion. Where it originates, where it inserts. And some of these you know already. For instance, the temporalis. The name tells you it originates from the temporal bone. Where does it insert? It inserts upon the coronoid process of the mandible. So if you remember your bones, it's going to make this much, much easier. You'll be expected to know the function, okay? And then you'll be expected for some of these to know the innervation. And again, you know many of these already. The orbicularis oculi is innervated by the facial nerve. The biceps muscle is innervated by the musculocutaneous. The quadriceps muscles are innervated by the femoral. Okay, so it's not terribly difficult. But the more you review on your own, this last part is an easier test. The difficulty is just the names and just remembering origins and inserts. It's not difficult concepts. It's just pure memorization of details. And some of them you've been exposed to already. Some of them you already know. So let's take a 5-minute break, and then we'll come back. We'll do some microscopes. We'll look at smooth muscle versus cardiac versus skeletal.